Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen, and welcome to the guest lecture sessions for the 19th Intermedical School Physiology Quiz or IMSPQ 2023. Would like to good morning to all of the participants, our honorable guests, and also all the participants who attend offline in Auditorium Gedung Pasasdana Tahir, Faculty of Medicine, Public Health and Nursing, Universitas Gadjah Mada, and also our friends, our participants who join online using Zoom, and also we have the live streaming on YouTube faculty at FKKMK UGM officially. It's an honor for me, Yopi Darwan, to host this visiting professor for the beautiful morning for the Friday, July 28, 2023. And for today's session, we have the lectures from one of our college from East Carolina University School of Medicine, USA. He is Professor Robert G. Carroll, Ph.D. He is also the expert in physiology. The session will be conducted until 11 a.m. So for the participant, we would like to invite to all participants who attend online or offline to actively give the discussion or comment to our speakers. Ladies and gentlemen, and to lead this session, I would like to introduce you to one of our lecturers from Department of Physiology, Faculty of Medicine, Public Health and Nursing, Universitas Gadjah Mada. She is Dr. Denny Agustiningsih, MKes IFM IFO Consultant. And let's take a look for her short profiles. Committee, please show the Dr. Denny's profiles in the screen. And let me introduce you, Dr. Denny is Associate Professor of Physiology, and also she is the head of the Medical Study Program at the Faculty of Medicine, Public Health and Nursing, UGM, and also she has an expert in clinical exercise physiology. So if you have questions or comment, just type your question or comment in the chat box in the Zoom, or participant who attend offline, you can raise your hands in the end of this lecture session. And for the next around 60 minutes, I will give the stage to Dr. Denny.
Thank you, Yopi. Assalamualaikum warahmatullahi wabarakatuh. Morning, ladies and gentlemen. Good morning, our distinguished honor guests, Prof. Carroll and also Prof. Cheng, and also the guests from the Zoom that join online. Today we will hear the lecture from Professor Robert Carroll about the calcium homeostasis. Professor Robert Carroll is Professor of Physiology. He is Associate Dean for Medical Student Education in East Carolina University, and also in 2018 he is a distinguished teacher from Robert AMC AOA Robert G. Glaser, and right now Professor Carroll is a chair in the IUPS Education Committee. There's a few books that Professor Carroll has written as an author. The interesting book is one is Integrated Physiology and one is in PPL, because Professor Carroll is also expert in medical education. Professor Carroll has a lot of research publication that in physiology, mostly about the long-term regulation of blood pressure, either in human or animal. One of the interesting topics is his PhD, that his research is about the long-term regulation of the blood pressure in shark. Not only human, but also shark. Yeah. Okay, for the about 90 minutes, we will hear and study with Professor Carroll. Times is
Thank you for that kind introduction, Dr. Denny. I appreciate the opportunity to come here. I congratulate the head of the department and his team for hosting me, Professor or Dr. Rahman, Dr. Jarjar, and it's been great to come here and to have a chance to get to see the campus and also to sample some Indonesian food. I'm wearing a tie with Tabasco sauce on it, which is a very spicy sauce from the southern part of the United States, to help protect me against the spiciness of the Indonesian food. So, assalamu alaikum, namaste, good morning. I think that covers most of it. Selamat pagi, of course. So my talk today is going to visit the homeostasis of calcium in the heart. One of the premises is that students don't learn from lectures. Students learn from stories. So if you're going to teach something, you need to provide the context, you need to provide the background, the story, and then tell them to memorize the important parts afterward. But it's much easier for them to remember it if it's told in a story. So today, I'm going to tell you the story of calcium homeostasis in the heart. Our understanding of that has evolved as we went through. As you look at this slide, shows the worked. So this uses a fluorescent dye to show the spike in calcium that happens in cardiac myosin. So we're going to come back to this slide at the end of the talk. And then when you look at it again, you have a deeper understanding of everything it is that you're watching, that small video. As with all things, it's important to behave ethically. So beginning a talk, you indicate whether you have a conflict of interest. I have no conflicts about today. So the first slide is I also need feedback from the, I would like you to join at the Menti code Menti program. And while you log on,
All of the participants here offline also join this Mentimeter, so we can give feedback. From the Zoom room, is there any difficulties to join the Mentimeter? Okay.
very good. So first question is, where are you from? And this is because I'm old. I have an inherent distrust of technology. I'm actually still surprised when technology, instead of being frustrated when. So I like to begin by identifying where you're from, just to give me a level of comfort technology is working. Okay, so most of the participants are from Southeast Asia, but we've got a couple from the South Central, and so that's a good result. I now have confidence in the tech. back to the PowerPoint. I've been very fortunate in my training. In my postdoctoral training, I got to go to the University of Mississippi, work with Dr. Arthur Guyton and Tom Lohmeyer. I spent 3 years down there, and it was an exceptional training environment, mostly because it was academically honest. Or
Next slide.
Yep. So it was academically honest. You changed your belief based on where the data would take you. So if the data do not support the theory, first check the data. Don't trust the data when it first comes out. But then if the data are valid, change the theory. So what we're going to talk about for calcium regulation, how our theories evolved based on data that's been collected, how we've had to change things accordingly. So we should have another Menti question. So what solution will best support the isolated part? And majority of people seem to be favoring the distilled water with some electrolytes added to it. So we'll come back to this question a little later on. For our first story, we're going to talk about Dr. Sidney Ringer and Mr. Fielder. The research setting for this was at the University of College of London, 83, so 140 years. What Dr. Ringer was doing was he was looking at the impact of changing ion composition on the perfusion solution for both skeletal and cardiac muscle. What he found out was that it was essential that sodium and chloride part that perfusion solution. However, one day when Mr. Fielding was sick, Dr. Ringer went into the lab, did the experiment. When Dr. Ringer mixed the solutions, the preparation looking at skeletal muscle function worked fine. Preparation looking at cardiac muscle function stopped working. Next day, when Mr. Fielding came back, both the skeletal muscle and the cardiac muscle worked for over an hour. So, created a problem. Why was it in why did a research outcome in one person's hand yield one result, in another person's hand a completely different result? Same lab, same physical space. So what they did was they found out that when Dr. Ringer mixed his solution, the heart stopped functioning. So back to the basic premise. The data do not agree with theory. First check the Dr. Ringer mixed his solution starting with distilled water, where Mr. Fielding mixed his solution with tap water, faucet, lab. And in 1883, the London tap water contained a number of other salts, including calcium. So, what conclusions do you draw from experience? Still care, because clinically, asked to give infusion solutions to patients, you'll either be giving lactated ringers, named for Dr. Ringer, or 0.9% daily. So even 100 later, still some argument as to which will be the better solution. If you're on a cardiac care floor, they tend to like the lactated ringers as a solution. Most other floors in the hospital, isolate 0.9% daily. So the conclusions is the first, never let the big boss do the experiments. Anytime the big boss comes into the lab, bad things happen. Anybody who's been a graduate student or research tech. However, the other conclusion was that extracellular calcium absolutely required for cardiac muscle, but it's not required for total muscle. Even though they're both striated muscles, the way in which the contraction occurs regulates very differently at cell level. So now we've got 3 mentee questions. Last one. The repeat question. So now when you're looking at solution that you would want to choose if you're going to do research on the isolated part, now the tap water would have the extra calcium in it. One, and change in your thoughts from what happened. So I'm feeling better that you are actually awake, listening as I'm talking. The next mentee question. So in contrast, if you're looking at a piece of skeletal muscle, the gastrocnemius muscle, which solution would be preferable? And for this one on an examination, it would be a bad question. I would have to accept 2 answers. Both the tap water or the distilled water would work equally well in terms of supporting skeletal muscle. So we've established that extracellular calcium is essential for the functioning of cardiac muscle. Next mentee question. Projecting forward onto the next topic, how would you have drugs that are selectively able to block L-type voltage gated calcium? And how would giving a drug which would block that, for verapamil, example of that, how would that impact the force of muscle, force of contraction of muscle cells? So second choice is decreased myocardial force of contraction. Got an increased myocardial force of contraction. Between the group, increasing the myocardial force of contraction is winning. Let's examine that premise and how well we come. PowerPoint. So, in moving to the second story, We're looking at the pathway for calcium entry in myocardium, L-type channels. So when looking at, physiologists like to group things into like types bucket. Lets us identify shared in common. One group of channels are voltage gated, open or closed on the membrane potential. Second group of channels are ligand gated, have a drug, agent, a chemical, hormone, some compound binding to the channel, then that will open or close. And so finally, some are both gated. So when we'll touch on the sinoatrial node funny current carried by the hyperpolarization activation cyclic. So both membrane potential and a ligand alter those. So for voltage gated channels to work, concept that you need to identify is the concept of threshold. When the membrane potential changes from below threshold to above it, from above threshold to back below it, causes a conformational change in the membrane protein structure. So that it's just a small rotation. That small rotation is enough to open or close channel. So channels usually can only get through a selectivity filter in the middle. So we're going to spend, or a lot of the focus is going to be what is threshold, how does change membrane past threshold impact performance. So this is an animation done by a friend of mine, David Brown, who was teaching cardiac section of the course at one time, and he had better animation skills than I had. So it was simpler for me just to steal his animation than to try and do a very bad reproduction of it on my own. When you look at this animation, got a membrane potential. Top of it is the extracellular surface, bottom of it is the cellular. The transmembrane spanning channels. There are two structures which can block channel. Usually the sodium ions, which are shown in green, can't get through. So at rest, there's a large driving force, but there's no pathway for the sodium to get through. So the sodium can't tell channels. Now, once you depolarize the membrane potential to 50, channels start to move. One on top moves very fast. The one on the bottom somewhat more slowly. So back up again. I'm going to depolarize to 50 and look at what happens. So the gate on the top pulls back very quickly. That allows the sodium channels to come through. Then the bottom gate closes off. So you have a period of time where sodium is permeable cell, then that closes. When you repolarize the membrane, drop the membrane potential back below 50 millimeters, the bottom gate swings out of the way early, and the top gate closes quickly. The bottom gate swings out more slowly. Never have any sodium permeability polarization. So now we're going to introduce another channel. So we've got the sodium channel on the left. We're going to introduce the potassium channel on the right. The sodium channel, the gate on top, the activation gate moves very quickly. Inactivation gate, the ball and chain on the bottom moves more slowly. And on the potassium channel, the end gate, slowest of all. So the red dot on the bottom lets you track the membrane potential. Membrane potential goes along, both the sodium and the potassium gates are closed. However, once you cross threshold, all three gates are at the same time. First gate to move is the activation gate. That lets sodium come in. You have a sequence of times where the sodium channel opens and closes, and finally the potassium channel opens. As you're looking at the membrane potential, the membrane potential gets to minus 50 millivolts. All three gates start to move. Have a period of time where the sodium permeability is increased, then the sodium closes. Potassium permeability is dominant. You get a hyperpolarization end that. Once the hyperpolarization finished, cell is out of its relative refractory period. Go back to excited. So the behavior of those channels differs by tissue. You look at the action potential for an axon, it's short, it's only about 10 millimeters. You look at the action potential for a piece of skeletal muscle, it's going to be somewhat longer, 20 to. You look at the action potential for cardiac muscle, about 200 millimeters. So that potassium channel for the heart doesn't open until 2 seconds after you cross the threshold. There's a large time delay where that potassium channel by the membrane resting. So in looking at the L-type voltage gated channels, that those channels mediate the cardiac but not skeletal muscle contraction. Slide shows the tubules, the imagination. The orange channels are calcium channels, voltage gated calcium channels. Then the green transporter down on the bottom, sodium calcium. So what happens, the way cardiac muscle works, calcium has to come in through that voltage channel. Calcium comes into the cell, binds with the ryanodine receptor, sarcoplasmic receptor. Then that opens calcium channels on the SR, so you get additional calcium release. Talk about the calcium coming from outside as a trigger calcium. Then the remaining calcium is SR. So about 10% that rise in calcium coming from outside the cell, about 90% coming from the sarcoplasm. Once that calcium levels increases inside the cell, then it's going to bind to the troponin C, and that will set off the sliding cross bridge filament mechanism. So important to recognize down on the very bottom figure on this, sequence of events. First, you've got the action potential, then you get a calcium spike, and finally, you get the contraction. The electrical has to be mechanical. Action potential absolutely has to happen. You're going to get a contraction, cardiac muscle. So, point, not quite. Why do you care about this? When you look at the Wiggers diagram, still have the electrical preceding mechanical. Look at this figure, QRS of the electrocardiogram precedes the pressure development in the left ventricle. So first you have to depolarize the ventricular muscle cells, calcium, then do you get the. The other reason I put up this slide, that when you look at the very top of the pressure tree, aortic pressure wave, Ph.D. mentor worked with Dr. Wiggers, part of his postdoctoral, and his job was to establish the relationship aortic pressure wave, ventricular pressure wave. So that top of that curve is part of his postdoctoral work, trying to figure out how the entire cardiac cycle works. My mentor's name was David Updike, and he was also the founding chair for the Department of Physiology via. So started my string of connections. So now we have another two mentee questions. Force of contraction. Majority of people are happy that it's finished whenever you give a calcium channel. Next question. Feeling much better about myself. Everybody seems to be liking the correct answer. Next. So, how will an increase in intracellular calcium affect the contractility? Now we're only one beat, single isolated. If you have an increase in calcium, how is that going to alter contractility? Seem to be running about 2 to 1 or 3 to 1 favoring an increased contractility. So let's go back over the data and see how that. So the first thing we have to establish a common set of definitions. One of the points of confusion for our students is contractility and those with characteristics. Contractility, not. So you have a challenge when looking at the heart. Skeletal muscle, doing work in a research lab is pretty easy. A linear structure, you tie the one end of the tendon to transducer, the other end weight, measure force development, traction, because it's a linear system. Trouble with the heart is cardiac muscle fibers are not arranged in a line. They're spiral shaped, wrapped around the heart. That's tied to their function. Cardiac muscle fibers aren't there to shorten. Cardiac muscle fibers are there to size chamber. So they've got to wrap around that chamber. Contract causes a chamber size. That gets to be a problem for a research lab. Skeletal muscle, it's easy to do the preload, afterload, all those kind of things. But for cardiac muscle, more challenging. But there is one piece of cardiac muscle arranged in fashion. The papillary muscle, part of the myocardium holding the edges of the cardiac valve leaf, are arranged. So other than that, they work just like cardiac muscle. They are myocardium. Now you've got one that will match research lab have for skeletal muscle. So the information we're going to talk about now refers to what happens in capillary experimental preparation. So I also like teaching by analogies. Greek warships, triremes, had 3 sets of oars. If you look at the very front of that, right down at the water line, a really sharp end. The way wars were fought, get the boat up to speed, then you ram another boat down at the water line, creating a hull so that the boat would flood, sink. So if you want to go fast, you got 2 different options. One of them would be to get more oars in the water. That's preload. Overlap of the actin and myosin filaments, anatomical relationship, where when you stretch the muscle to length, you get the maximum overlap of filaments. Skeletal muscle only has preload available to option. Cardiac muscle has another trick. Cardiac muscle can change the rate of ATP gaze, driving the contraction. So when you look at the ships, there's usually some guy beating a big drum on the back of that ship, keep cadence. So everybody is rowing in unison. If he picks up that cadence, the boat will go faster. So that gives you a second way to get more oars in the water, or you can have the oars that are in the water cycle more. Doing that, you get a stronger force of contraction. So, turning to the question on calcium preload and afterload. Capillary muscle. Now we're up to a 4-to-1 or 5-to-1 favoring contractility. Correct answer. Calcium is only involved in contractility, resting length, that just by the mechanical structure, how stretched the filaments are before they begin. So moving on to the third part of the story, calcium and myocardial contraction. Beta-1 adrenergic receptors increase contractility. You have an increase in heart rate. That's also tied to an digitalis is a drug, a cardiac glycoside, digoxin, and it can be. Look at all 3 of those things, what they have in common, all work to. Calcium is going to be driving the myocardial contract. It wouldn't be a physiology lecture if we didn't have second message. So on the top, the beta-1 adrenergic receptor will bind both epinephrine and norepinephrine. And then it will set off a series of events leading to the activation of adenylate cyclase. And Cyclic AMP going to work to activate protein kinase A, and that will phosphorylate that voltage-gated calcium channel and the ryanodine receptor. Those guys step up their level of activity, phosphor. Work from David Beers, published in a Nature article, and it's a really great summary of all the events time. So, what happens if you have an increase in heart rate? How does that influence myocardial contractility? At one point, this was the prevailing theory that if you have a normal heart rate, cells depolarize for this period of time, how much calcium gets. If you have a faster heart rate, increase in heart rate comes at the expense of diastole. The heart stays in polarized state more, and you get more calcium. So there was a net increase. Thought that there was calcium gain just as a virtue of that fast. You look at how this plays out in humans, when you look at the cardiac output and the maximum heart rate, the stroke volume goes up very quick. Cardiac output, the heart rate goes up in a more linear fashion. So by the time you get out to about 15 liters per minute, about half maximum cardiac output, exceeded the capability of preload to help you system. Rest of the improvement, cardiac output, has to come heart rate. So now we've got to take a side trip in our story. Those stories are ever linear, always these. Myocardial cells, notably the pacemaker cells of the SA and AT node, spontaneously rise to threshold, will increase, cause action potential. That's referred to as automaticity part. Early depolarization, mediated by the hyperpolarization and changes of the potassium channel, closing of the sodium channel. Now, the researchers on this called it a funny current because every other voltage gated channel was closed by hyperpolarization. The sky was opened by hyperpolarization. And the name stuck. This is what they just called it in the lab when they were complaining about it. There's that funny current again, and now called the funny current. The later stages of spontaneous depolarization are mediated by the L-type calcium. So the reason that we're going through this diversion, because I can't talk about myocardial calcium without at least acknowledging the as. So here you see some Purkinje fibers, and Purkinje fibers can take over as pacemakers if the heart rate is down to 30 beats per minute. And you see the calcium spikes spontaneously increasing, so one of them depolarizes enough, and that wave of depolarization entire myocardium. So heart rate is part by calcium type calcium channels. That also means a slower heart rate, one of the consequences of rapamycin drugs that block. So moving on to the next topic, cleaning up your room, putting things back. Your mother told you, finish playing with something, put it back. And all this time releasing calcium, now we've got to put it back where it. So for the calcium release, it was easy. All you had to do was open a channel. Radiance always favored calcium moving from the storage areas, either from the extracellular into the interior of the cells or from the sarcoplasmic reticulum the interior of the cells. The massive 1,000-fold. So getting it out, no problem. Putting it back, you got to move an ion against its concentration group. That means it costs. You got to spend energy. So now you start looking at primary active transport, where you're going to have a calcium ATPase, or secondary active transport. You're going to use the sodium gradient in order to drive the calcium. One of the hallmarks of medicine that when we look at traditional medicines, gives us windows to how things actually will work in case you're doing experiments. So the foxglove plant used to produce a drug useful to heart failure. And they purified this. They found out the digitalis cardiac glycosides was active in foxglove plant. So when you give the digitalis, it blocks the sodium potassium ATPase. So now looking at the top of this slide, on the top, have ways to get the calcium that is inside of the cell, pick it out of the cell, move it back to the extracellular space. One choice is a calcium ATPase. Second choice is a sodium calcium exchanger, where you've got to bring in 3 sodium, 1 calcium across, because that's how large the ionic imbalance is. Calcium makes the energy from 3 sodium drive 1 calcium. Then finally on the top, you have the sodium potassium. Sodium potassium ATPase is the target for the cardiac glycoside. What happens is poison that pump as you finish the effectiveness. Then potassium stays extracellularly. Sodium does not stay. Increase intracellular sodium, extracellular potassium. As sodium builds up inside of the cell, no longer a sodium gradient. So that 3 sodium trying to come in for the calcium exchange is not going to work as well, because it was dependent upon the sodium gradient. So the consequence of that is intracellular calcium levels increase, and that leads. That's how the digitalis is work, enhanced cardiac ability. Now, a lot of countries, a lot of medical groups are recommending against digitalis because it's got a very narrow therapeutic difference between something that is helpful for a patient in heart failure, something that will kill them because of the hyperkalemia, a really narrow window. So there are better drugs available now to assist in. So when you have the cardiac relaxation on the endoplasmic reticulum, you got the calcium ATPase. That calcium ATPase is regulated by a protein called phospholamban. Probably the most fun protein name to say. So when you look at it, phospholamban, when it's unphosphorylated, is a brake on the system that slow down the calcium. Phosphorylate it, you pull it off and you get rid of the brake, accelerates sodium potassium. So that way you have the ability to regulate how well that calcium sarcoplasmic reticulum is going to function. Question for Menti? One of the, wait, only about 4,100 1,000 milliseconds, 400 milliseconds, called an extra. Got the pressure development will increase because of high calcium. About 2 to 1 over pressure development will decrease because of low calcium. Back to the So the post-extrasystolic potentiation. You look at the premature beat. This is an electrocardiogram tracing. So the arrow on the bottom is at the ventricular extrasystole. And then there's a pause because the electrical events happen in the ventricle. So the SA node is still firing at its regular frequency. So there's an extended pause before the next beat. So the premature beat originates at the ventricle because the SA node fires irregularly. Premature beat with a short interval, followed by the extrasystole, a much longer than normal. Together, they add up to. So now let's look at the data show. For the post-extrasystolic potentiation, got the kg, pressure development, and then the rate of pressure development, the first derivative on the pressure development. And if you look at the marking on this slide showing the extrasystole, it's got a wider electrocardiogram of that, not using regular conducting tissue. You look at the left ventricular pressure development, greatly diminished. Look at the rate of how quickly that pressure is developing, diminished even more. If you look at the post-extrasystole, the electrocardiogram for that looks relatively normal. But when you look at the pressure development, it has a higher pressure development. In particular, that when you look at the rate pressure development, contractility of that. So the premature ventricular contraction results in a decreased contractility and pressure generation. Subsequent heat delayed, that's the one that shows the contraction. So the thought is, well, it was just because the ventricle didn't have enough time to fill. That's why you're not getting that pressure development. They took the data and they went into the papillary muscle preparation. Same results were. So it's not ventricular filling, it is a function of muscle. So the findings, a fast heart rate causes an increased contractility, and early extrasystole has the same timing as that fast heart rate. So if the data doesn't agree with theory, first check the data. Studies with the papillary muscle showed a weaker contraction, faster pacing. So if the data do not agree with theory, first check the data. Data are valid, change the. So we've got to go back to the sarcoplasmic reticulum. Not as simple a story as we. What we now know is that, and this is from Aghai et al. 2014, that when the calcium is pumped back into the sarcoplasmic reticulum, not the portion of the SR where it gets released. So there is a SR processing of calcium, binding the calsequestrin, so enriched calcium binding protein. Then it has to be carried over to the part of the SR where the ryanodine receptor is used. Happened for the early extrasystole, the calcium had not migrated the portion of the SR from which it is going to be. opening a channel did not give you. For the post-extrasystole, calcium now has all had a chance to get up to the portion of the SR where it's going from. So the post-extrasystole actually enhanced calcium during the polarization. So why is the rapid heart rate tied to an increase in contractility? Because I thought that was because the calcium. And what happens is when you look at the left-hand side of this figure, it was all the things that adenylate cyclase did, cyclic AMP did to change the function of the anodine receptor calcium channel. On the right-hand side, you have the things that the calcium or that the activation of beta 1 adrenergic receptor does change how calcium is handling. So what happens is activation of the beta adrenergic receptor ultimately leads to phosphorylation, that phospholambam. Pull off the brake. So the calcium is able to put back sarcoplasmic reticulum quickly. That's what gets it up to the end of the SR where it's going to release. So if this didn't happen, then the increase in heart rate not be tied to an increase in contractility. Whole calcium handling dynamics have to change in order to accommodate contractility tied to calcium, tied to elevated heart rate to one. So returning to last, I believe, of the mentee question. Question is, how would the pressure development compare to the following the contraction post-extra? We're running about 3-to-1 in favor of the answer that I like, which is the extrasystole would be weaker and the post-extrasystole stronger because of how the calcium dynamics have changed, how handling the. So now we've got the summary figure. Got enough information that calcium story together. I like this figure because it has all of the calcium release. Left-hand side of the figure, all the calcium concentration, right-hand side. Taking through the sequence of events, for number one, you've got to get the depolarization myocardial. Polarization is going to open the gated calcium channels. Extracellular calcium is going to come in, bind to the ryanodine receptor on the sarcoplasmic, cause calcium, additional calcium. Combination of the extracellular and the SR calcium, both going to join to cause the actin and myosin to bypass each. Shortly thereafter, going to put all the calcium back to where it came. Calcium ATPase is going to pump it back into the sarcoplasmic. On the extracellular side, most of the calcium is excreted through that sodium calcium exchanger. That's how you get the 10% outside of it. Put it back the outside of this. So again, why do you care about it? Where's the clinical application? There are a number of proteins that are involved in the SR handling of calcium. As we're looking at heart failure is now being split into an ejection fraction diminished, ejection fraction preserved heart failure. So for the people who have the ejection fraction diminished, that's the contractility problem. Heart is not working as for the ejection fraction preferred or preserved heart failure. For those individuals, it's a relaxation, not getting the heart to relax because of the abnormal calcium rate. So there's a whole lot of research now being done on the dynamic or the mechanism changing sarcoplasmic reticulum handling of calcium, looking to that as venues to try and improve patients who have the ejection or preserved heart failure. And so now the story closes. You look at what happens. Depolarization is going to allow extracellular calcium influx to come in through the L-gated, L-type voltage-gated calcium channels. That calcium is going to bind to the ryanodine receptor, triggering calcium R. Cellular calcium binds to troponin C, ultimately leads to sarcomere shortening. Calcium is released from troponin C, put back into the SR through the calcium ATPase or across the membrane through the calcium exchanger. Calcium in the SR is processed, prepared for the. So when you look at this, less, there we are. So look how quickly that calcium is removed. Get that massive burst, then it's removed almost. So it's that calcium resequestration, how quickly that occurs, which is focus or mechanism behind cardiac. That, Dr. Denny, thank you for hosting me, and Terima kasih.
Thank you, Carol. You can sit and be entry for answering question. Thank you, and give big applause for Professor Carroll. Okay, today we very interesting, very clearly lecture from Professor Carroll about the calcium homeostasis. That we know that the calcium homeostasis is very important for the cardiac function, not only for the contractility, but also for the electrical activity. And this calcium will underlie the lot of pathophysiology of the cardiac diseases. Professor Carroll already gave us insight about the calcium handling, that and also we have a new insight how the interaction between contractility of the cardiac muscles and also the preload, and also for the other muscle function. And for this lecture, as the teacher, we also learn that when we want to give a basic concept for the students, we have to emphasize the story behind the concept. So this concept will more understand. Okay, I think a lot of interesting lecture for this Professor Carroll. So I believe there's a lot of question for the audience. Before that, my thesis, my PhD thesis about this calcium. I block the L-type calcium cardiac muscle.
Then
But
you
in
should have
that
been giving
way,
this
you know,
talk.
that's why it's very interesting topic for me. I think it's also for the audience. So we can open for the questions. Maybe we have 3 questions for the first round. Okay, 1, 2, Dr. Rahma. Once more, there. Once more, maybe from the Zoom room. Is there any question from the Zoom? I have the committee can write down the question from the Zoom room, or they can raise their hand. Okay, we start with the gentleman there. If please state your name and where do you come from?
Okay, thank you for the opportunity. Good morning, Mr. Robert. Thank you for your presentation. Thank you for the change me asking this question. My name is Rizal. I'm from electrical engineering, so that I don't have really good basic, but I'm interested to attending this uh, topic. I have a question about uh, i- to detect the amount of calcium in our blood. Uh, there's some tests, or I mean, instead of that test, can we also detect it by a picture of heart? I mean, the scan, so that we can early detect uh, the further, maybe the disease that we can get from it. Because uh, there's some a technology that we can detect, like from machine learning, something like that. If we can detect it earlier, we can know how to prevent it.
Thank you for the question. Calcium serves a number of roles in the body. What we were focused on in this talk was intracellular calcium and its impact. However, plasma calcium can be involved in the creation of atherosclerotic plaques and other problems like that. I know there is a calcium scan which is done on patients in order to look for the early development, accumulation of calcium, some of the myocardial blood vessels, one of the earlier signs for development or for plaque formation. And I need to say right up front, I have no clinical background. One of my colleagues had this done. And so we went and looked and saw how it was done and looked at the results and all that, because we like to find out why things are done. He didn't have any real problems. His scan came out very. So yes, there is a lot of work being done on calcium accumulation in plaques as an early marker for cardiovascular, particularly in myocardium.
This our friend from electrical engineering is develop many device scan to predict some heart disease or so. As Professor Carroll answered, we can scan calcium accumulation the blood vessels, but not in the cardiac muscles.
And part of that is the order of magnitude. Plasma calcium at the 1 or 2 millimolar range, I believe, where intracellular calcium is 1 nanomolar range, the micro and nanomolar. So an order of magnitude smaller, you wouldn't be able to see the cellular calcium levels because the blood calcium would so much more of it's in.
Dr. Rahma? No. Too clear for you.
Okay, especially in the person who suffer high blood pressure, also especially in elderly person, it's just out luck. It will influence in receptor. So the mechanism of control and communication will change. Yeah. You think about plaque.
And this would not only be the calcium plaque, but when you look at the signal for the baroreceptors, the aortic arch and carotid artery baroreceptors, they respond to stretch. So as the blood vessels become more rigid, atherosclerosis, then they don't stretch as well, and that was the signal the baroreceptors were looking for. So they actually think you're hypotensive because they're not getting receptors. Think you're hypotensive because they're no longer being stimulated, and that will then shift over to salt water conserving stance to cause a higher blood pressure. The relatively good news is the baroreceptors are somewhat short term. They adapt over time. So as they're great for detecting changes in blood pressure within seconds, within minutes, days, they start to become a little less so, weeks, less so. So they're great at short-term regulation of blood pressure, but they don't have as large an impact as long-term regulation of blood pressure. For that, the kidney is going to be focused.
Zainal, baroreceptors and calcium baroreceptors. Very important for regulation of the calcium. Okay, Dr. Dite, maybe.
Thank you very much for the. My name is Tito from Talk. Really interesting talk. And I have a several questions. The first question is, you said earlier there are many mechanisms of handling from the L voltage gated and then natrium K exchangers and then phospholamban, and then also there's this SRKA2 also that regulates the calcium intracellular. If there is some disease or some problem with the handling of the calcium in one, like in terms of the L voltage, there's a problem there, and it can cause the due to some problem with the depolarization. And then is there any like a component in this, and then phosphorylation will be it, and then calcium will is to compensate the problem in one part of the calcium handling. So this is my part of the that in kind of overexpress the this calcium handling mechanism in some part, in some drug or some therapeutic agent. I think. Thank you very much.
Thank you. Great question. Now, I'm going to go at it from the bench scientist perspective. The trouble with the intracellular calcium and using that as a driver for what will happen next is there's not a good sensor for what intracellular calcium is. So I have the calcium is responding to other agents like the beta adrenergic receptors, like Wabane, and not driving changes. So from a control system's perspective, it's not a regulated variable. And because it's not able to be sensed, it would be a poor candidate search. You're looking at will it drive the next set of. Now, next question about looking at the calcium dynamics, that's a very good area for research. And I have to apologize because I don't know where the current research is on this, but I know about 4 or 5 years ago, there were a bunch of pharmaceutical companies all racing to try and find agents that would be effective and specifically preserved ejection fraction heart failure, the diastolic heart failure. 10 years ago, there was no choice, there were no treatments for that particular type of heart failure. And they're trying to develop them. Calcium, altering the calcium handling more promising sides, but I've not paid how those studies have. Great questions.
Think, yeah, from the internal medicine. He already back from Nobel Physiology laureates Nobel Physiology in Germany. I think there's a lot of research in the calcium handling, you know, either in the animal or the humans. That's why there's a lot of drugs choice for this calcium channel. So that's why the lecture from Professor Carroll is very important, not only for our in the basic science, but also for the clinical scientists and also clinical practice, because when we have to choose which drugs is right suitable for the disease, is also depends on which calcium channels that we have to block, or when we want to regulate the contractility, because the calcium channel in the cardiac muscle is not only L-type cardiac channel, not only not the sodium calcium exchangers, but also there's a lot types of the calcium channels in the cardiac muscle tissue. Thank you, Dr. Dite. Maybe there is any questions, Dr. Mumtaz?
Thank you for Mr. Robert. My name is Mumtaz Maulana Hidayat. Call me Mumtaz. I'm coming from physiology. I'm still learning biomedical science. I'm now a general physician, and I really interesting about calcium channel blocker, because we know that in Indonesia, especially in primary healthcare, we as general physician uh, in some case using calcium channel blocker like lodipine, nifedipine together to treat primary hypertension. And we know that calcium channel blocker can block the calcium channel like L-type, as you explained before. And my question is, because we're using a calcium channel blocker in long term in some hypertension patient, can we, can we know that we have some potential long-term effect like arrhythmia or heart failure, because we sometimes using it from long time until the patient death. So can we have maybe the other option using other drugs, other better option instead of using calcium channel blocker? Because my opinion is, if we using calcium channel blocker for a long time, maybe from your explanation, we have some potential long-term effect to the rhythm and the contraction of the heart. This is my question.
And once again, I'm going to go back to the bench scientist approach to your question. In terms of what drugs to use for long-term management of hypertension, the focus that I would have is they're all going to be impacting the kidney one way or another. So because of that, if you would want to take a patient off of one drug and shift to another regimen, I would look for something else that is a diuretic, some other form of managing the fluid balance within the patient. Part of my postdoctoral work was, I said, in Mississippi with Dr. Guyton, and the party line for the department is the kidney is the only organ involved in the long-term regulation of blood pressure. And they've had all kinds of experimental data to prove it. So I would let you have to go back to your clinical colleagues to provide the better answer for that. But the researcher side of me would be, the simplest thing would be just to shift to some other agent which will impact renal function.
Professor Carroll, maybe we move to the next question from the Zoom room. Is there how many question from the Zoom room? Is there in the chat room, or for question from the chat room, or maybe some audience want to directly speaking? Okay. The first question from Nabil from Faculty of Medicine Universitas Muhammadiyah Semarang. Yeah. Thank you for your great explanation, Prof. I would like to ask two questions. One, what the best research method or modalities to know the effect of calcium homeostasis in heart functionally? Is it by patch clamp or surface ECG or by ex vivo? And then the second one, what is the role of CAMK2 protein in calcium homeostasis? Thank you.
Okay. For the first question, the research modalities depends on which question you're asking. So for the surface CKG, there's the consequence of hyper and hypokalemia and the ECG changes are pretty well established. So there's not going to be a lot of research around that topic. The patch clamp, if you were able to isolate the sarcoplasmic reticulum membranes and then do membrane studies on just the SR section of the sarcoplasmic reticulum membrane, that would be great. But technically, I'm not sure how effectively you can isolate SR from fractionization. The over and under expression of the proteins would be the better choice for trying to alter SR functionality. And then the ex vivo is probably going to be the most powerful. The fluorescent dyes, the Fura2 type dyes that I showed the images of, are all done ex vivo. In vivo, the over under expressions of the proteins would be about the only way that I could think of to ask the questions. And the role of the CAM2 protein in calcium homeostasis, I'm going to keep my mouth shut because I know just enough about that to be dangerous. And so I would have to read up more on that before I would be comfortable responding to that. Is there a relationship between calcium and mitochondria? There's a massive relationship between calcium and mitochondria. The article that I studied, or the last paper that I cited, the 2/3 of that paper were devoted to the altered mitochondrial function in the calcium regulation. I did not branch in on that because I was just trying to focus on the cardiac function. So yes, the mitochondria and the role of calcium in regulating energy production is a very, very active area of research. Some questions: is there correlation between osteoporosis in elderly with the calcium homeostasis? Might lead to heart failure. Again, no clinical background, but I would have a hard time drawing that link because the plasma calcium regulation is done primarily by PTH, our thyroid hormone, and the types of calcium deposition, getting rid of calcium deposited in the bone from osteoporosis is a different process from the PTH regulation of bone calcium. So I don't see how those two are coupled. I would have to see a disease process or an experimental model that would couple those before I would think that that would be a worthwhile place to do research. Can we use blood as a predictor of heart failure? Not really. There's plenty of calcium circulating in the plasma to drive the myocardial contractility. By the time you would drop the calcium low enough to impact cardiac contractility, you'd have a lot of other organ system failures. So a drop in plasma calcium, I don't see how that would be enough to impact the myocardial contractility. I would defer to the clinical colleagues to say whether or not that's actually true. Again, with the mitochondria, yes, as I said, the last paper that I cited in the presentation, about 2/3 of that paper was focused on the mitochondrial proteins that are altered by calcium dynamics. So that's a very active area of research.
Thank you. A lot of questions for clinical and also research, doing research with calcium topics. Is there any question from the Zoom room? Maybe we can add 2 or 3 more questions from this room if there's any. Dr. Adrianta?
Terima kasih. Thank you, Rob, for the good presentation. Once I emailed you, I'm as a teacher always have difficulty to teach the last part of cardiovascular physiology, that is regulation, isn't it? And you today mentioned kidney as one of the important thing to talk about that for regulation. And we don't teach that too much. We teach the RAS system, endocrine, adrenergic system, teach more about the acute regulation with the baroreflexes. So can you maybe explain more about, you mentioned the kidney is one of the most important one for regulation of the cardiovascular. As I compare to this regulation of respiration, it's easy for me because the concept is like there is mechanoreceptors to regulate, and there is the chemical one to regulate with oxygen, CO2, and pH. So it's more straightforward. For cardiovascular, it seems there is a system like the micro thing and the macro thing. So a little bit difficult to teach that topic. Can you make your lecture on that besides this calcium, if I may? Thank you.
Sure, in 1 minute or less.
30 minutes.
So during my postdoctoral work, one of the studies was on control systems, and then there's a concept of gain in a system. And that's when you move a system away from an equilibrium, how far the system gets moved back toward that equilibrium divided by the remaining gap. So it doesn't close it all the way. When you look at the cardiovascular regulatory systems and the gain of those systems, there are two perspectives. One is the pressure range over which they're acting, and the second is the time frame over which they're acting. So if you begin with the pressure frame over which they're acting, and you look at the gain for the baroreceptor reflex, it's massive, right around the 120/80 millimeters of mercury. If blood pressure gets higher than 150 or lower than 50, it's useless. It's already been turned on as strong as it's going to get. In contrast, the CNS ischemic response, the Cushing reflex, when blood pressure drops below 30, that becomes massively important, and it becomes the most powerful neural control. As we're sitting in this room, it's turned off. It's doing no good, no function at all. So what pressures or what systems you're looking at depends on what your starting point is and what your blood pressure is. The same way, if you look at the ones over time, if you look at changes in the baroreceptors over seconds, they're going to be a very, very powerful regulator of blood pressure. The renin-angiotensin system will become important if you look at hours. If you look at weeks, then the long-term regulation of fluid balance becomes the dominant. So which one is most effective depends on which time frame you're. Whether you want to look at an acute response, would be neural, a couple hours, which is going to be mostly endocrine, or long-term, which is fluid dynamic. 2 minutes.
Thank you for the tip. Maybe we can teach for this regulation not only in one topics. Maybe when we teach the kidney system, we give the long regulation. When we teach maybe previously for the cardio, we can give it in the acute regulation. Thank you, Dr. Adriana. Maybe is there any other question before we close this interesting session? Is it clear enough? I think because the Mentimeter also succeed to give the right, all of the participants give the right answer or have after the explanation. So I think all the audience is already clear enough, like Dr. Rahma says. We can close this lecture today, and we hope this what we get this morning will have a long-term effect of for our knowledge and activity. Thank you again for Professor Carroll for your nice, interesting lecture, and hope you can come back again to lecture us. Maybe tomorrow you still give us two kind of lecture. Thank you for your attention. Give applause again to Professor Carroll.
Thank you very much, Dr. Denning. Thank you.
I give it back to Yopi. Thank you.
Okay, everyone, once again, give applause to Prof. Carolina Sudotodeni for leading the visiting professor with the topic calcium homeostasis in the heart. So, Prof. Carol will be in FKKM game until Sunday, I think, or Monday? Monday, okay. So, if you want to have a long discussion with Prof. Carol, you can just ask him because he will stay in Jogja until on Monday. So thank you so much for the participant who joined online using Zoom. Can't wait to see you directly in Yogyakarta to join the 19 Intermediate School Physiology Quiz IMSPQ 2023 that will be held on a Saturday and a Sunday tomorrow. So thank you so much for joining with me. I'm Yopi. Sign out. Wassalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. And then we invite to all the participants who will attend offline to take a photo together. So please come closer in the middle of the seats. Zul or Mas Itam can help me. Prof. Carol, so you may back to your seat to take a photo. Mas Bulingin bisa merapat ke tengah untuk sesi foto bareng. Masih ada kursi kosong di baris kedua, silahkan. Zul mau ngambil juga? Okay, so